right, well, I won't give you a quiz then, since it's all that stuff. And I'll post a new schedule. Mostly, I think we'll just fix it. Does it make a test? I don't know. I can't read calendar. So next time we'll have a quiz on chapter 25, part 1.
Right. I didn't know. I need to look at this one, so this one is completely different. I was like,
I didn't even get that. So in this last time we went through kind of all the basics of the organ nucleotide tract, small intestine, large intestine, and most of today is going to be enzymes and regulation of all those enzymes. And making sure you're making the right amount of stomach acid or putting the right amount of digested food into the small intestine, the right speed and everything. So first, just take a quick review of how enzymes sort of work. This may be really obvious to some of you, but not everyone's taken chemistry or intro biology.
Is to use enzymes and other digestive chemicals to break down larger nutrient molecules into smaller absorbable molecules. Digestive enzymes do most of the work in chemical digestion. However, other chemicals are needed in digestion. They are water, bile, gastric acid, and bicarbonate. Chemical digestion consists of enzyme-mediated hydrolysis reactions. In this type of reaction, water is used with enzymes to break down a larger nutrient molecule known as the substrate into smaller nutrient molecules known as products. Enzymes are commonly identified by the substrate they break down. In this case, the enzyme dipeptidase breaks down the substrate dipeptide.
All right, so you may be wondering what water's there for, why you need it. And so basically most of what you eat are macromolecules. So it's a bunch of simple, mostly repeating units put together, like 1,000 glucoses attached to one another, or a protein with like 200 amino acids attached together. So what all these enzymes do is basically chop it into little pieces so that the small intestine and large intestine can actually absorb those small pieces. Your intestine can't absorb starch. It's too big, it would just kind of flow on through. So instead you gotta chop it up. So here's like your starch with some 2 glucoses bound together, and you've got some kind of enzyme in there. And you add water, which remember is oxygen, 2 hydrogens, and it's hydrolysis, which means the water splits, like hydrolysis. So the glucose, this bond gets broken, 1 glucose gets an OH put on it in the process, and the other glucose gets a hydrogen put on it. And now we've got individual glucose monomers that we can absorb. So same idea for all the macromolecules, the nucleic acids, the proteins, the lipids. Gotta kind of chop them up first. So you can imagine you need lots of different enzymes to do this. And that's the most important job of the pancreas. What's the other job of the pancreas that you've probably heard about? So like insulin and glucagon, blood sugar levels. only 1% of the area of the pancreas does insulin stuff. The rest of it, 99%, is all about digestion, which is interesting because most people don't know that. You just think of pancreas, you think insulin, because that's what you hear more about. But it does make tons of different enzymes. And of course, those different macromolecules, they have different shapes. So you need different enzymes that are going to recognize those different shapes to cut those molecules up. So it makes enzymes that break down carbohydrates, proteins, fats, nucleic acids. And a second thing it does that we'll talk about a little later is it makes sodium bicarbonate, which I think I mentioned last time. So the stomach acid with the food, it's coming into the small intestine at like pH 2 or 3. So right at the beginning, you put a bunch of bicarbonate, which is a base. Of course, you use it in cooking, and you know it as what? So baking powder, sodium bicarbonate. Or is it baking soda? I always get the two mixed up. I think it's baking soda, actually. What? I think it is soda. And I actually just found out something interesting about baking soda is that if you put it in beans when you're cooking them, they cook like in like half an hour instead of having to cook them for like 6 hours. Because it's a base, so it denatures the proteins faster. It's kind of interesting. Has anyone heard of doing that before? It's kind of interesting. All right, and it does put all its enzymes and bicarbonate right into the duodenum, right at the very beginning of the small intestine, which makes sense. You've kind of done the mechanical digestion, broken up the food into pieces, maybe you denatured some of the proteins with the acid, and so now we can get all the enzymes to really start chopping everything up. Right? And so it's kind of interesting. It's one of the harder organs to actually find when you do a dissection because it's not like a big chunk like the liver. It's sort of thin and sort of spread out against the small intestine. So, I mean, you'll see it when we do the dissect the cat. Is that next week we're dissecting a cat? 2 weeks? Yeah. So it'll be fun. All right, so it's on here. All right. So here's the list of all the enzymes you have to know. And there's a trick to these, mainly that their name tells you what they do, or at least a lot of them. So the easiest way to do it probably is memorize that amylase breaks down starch. And if you notice, do you remember hearing about amylase already? Where else do you make amylase? So it's in your saliva. It starts it, but the stomach acid destroys that enzyme. So you don't have any amylase once it's in the intestine, so you got to make some more. And it's a slightly different enzyme. And all of these, trypsin, chymotrypsin, elastase, carboxypeptidase, they break down proteins. And you need lots of different ones because proteins have all different shapes. So you need a bunch of different enzymes to break down the proteins in different places. Lipase breaks down lipids. Ribonuclease breaks down RNA. Deoxyribonuclease, what do you think it breaks down? It's just the DNA. So that's all the stuff that's in the pancreatic juice. All right, what if you're obsessed with whipped cream and you just eat a big pile of it? Unsweetened. I know. Hey, maybe you're on the Sugar Busters diet, I don't know. Well, looks like it's a battle between C and D. So you chat with your group there and see. So most people switch to C. I'd say that's probably the best answer. Do you think maybe pepsin could be right too, though? Yes. Right, because there's fats in milk, but there's probably also protein there. Which is funny because this is one of the, somebody trying to get in? No. This is one of the few like book clicker questions that I actually used. This doesn't even have a right answer. So really C and D would be right then. I don't know, maybe I should use butter. Does butter have protein in it, or is it just fat, do you think? It's just fat, right? I'm going to change it to you eat butter by itself. Like my daughter. You put buttered toast, and she just takes the butter, and she's like, mm-hmm. All right, so next up is our liver and gallbladder. So one of the things the liver does is make bile, which is a fat emulsifier. And we're going to watch a little movie about that in a second. And the gallbladder actually stores extra bile, so that you've got, a big pile of bile ready. If you eat something that's very fatty, then the gallbladder can just inject it right into the small intestine and get it all emulsified. So let's see what emulsify means exactly. So I found people have a hard time with what it means exactly. All right.
If you were to put some oil and water into a jar and shook them up, the oil and water would mix together and form a cloudy looking liquid. But if you were to leave the jar to stand for a few minutes, the oil and water would begin to separate. The oil will rise to the top and form a distinct layer on top of the water. Oil and water do not mix together permanently. But sometimes in cooking we do want oil and water to mix, and we can do that by adding an emulsifier. An emulsifier encourages the oil and water to mix. This is what is done in sauce recipes such as hollandaise sauce and mayonnaise. The emulsifier that's used in these recipes is called lecithin, which is found in egg yolk. This is how an emulsifier works. Emulsifiers are molecules with two ends. One end is attracted to water, it's hydrophilic, it loves water, whereas the other end is attracted to the oil. This end is hydrophobic, it really doesn't like water. When an emulsifier is added to a mixture of oil and water, its molecules arrange themselves so that they prevent the oil and water from separating, like this. The mixture has now become an emulsion. Emulsions are either oil in water or water in oil.
Trying to stop it on that part. So there's the little lipid molecules, and then the emulsifier has half of it that's attracted to the oil, and the other half is attracted to water. Do you know any other examples of emulsifiers besides egg yolks? Is there anything that helps get grease off of things or oil? Soap, right? So you got a bacon pan, you put it in water, and it just kind of sits there, it doesn't get clean, and you just put a couple drops of soap in there, all of a sudden the oil disappears, right? It's not disappearing, it's just getting into much smaller droplets, basically. So, yeah. So bile salts are emulsifiers. Why do you think you need to emulsify those fat droplets that you ate when you took that spoonful of butter? So that's what people would usually say, but you don't actually break any chemical bonds with an emulsifier. What are you doing to it? Closer. What? No, it probably wouldn't have trouble moving. What do you think would happen to this big bubble of fat that you just ate once you put emulsifiers with it? Okay, so instead of, we'll just put little molecules in here. Instead of a big, huge bubble, now we're going to get lots of little bubbles with just a few triglycerides in it. And this is important because what do we have to do to these triglycerides before we can absorb them? Can you just absorb them, or do we need to chop them up or something? And the enzyme that breaks down lipids is a lipase. So now that it's in small little bubbles, that lipase enzyme can find it and quickly break it into tiny little pieces. If you tried to skip that step, it would only be able to grab the triglycerides that are right on the outside of that bubble, and it would take a really long time to digest it. So it's the lipase there.
So isn't there an intestinal disease that doesn't produce that step?
Sorry, which of these two steps? The emulsifier step or the lipase step?
Emulsifier step.
Well, one example for people have their gallbladder taken out because it gets infected, inflamed, diseased. And so they only can produce small amounts of bile from the liver. They don't have that big amount they could put in. And so they can't eat as much fat. And they'll actually feel really sick if they eat ice cream or something really fattening. Actually, I had a student last semester was gallbladderless. Actually, I think two students. So, yeah. And I'm sure there's genetic things you can inherit where you don't make proper lipase or you don't make good amylase. Certainly true. And maybe you wouldn't even know. You just think you don't digest certain things well. Oh, and a fun thing you'll see in the dissection, bile is very green. And anybody know where bile comes from? It's a breakdown product also. Probably wouldn't know this yet. So it comes from the breakdown of hemoglobin from red blood cells. But you won't need to know that yet. We'll do that when we do the blood chapter. All right. So why is it important to emulsify lipids? All right. So A, B, and E. So if you chose B, you'd be correct. And I don't know why this always confuses people. This is something that people are always getting confused on. So for A, emulsification does not break the lipids into fatty acids. That's the lipase that does that. That's step 2. So step 1 is break the big bubble into little bubbles, which is not making fatty acids. And that does increase surface area, which is B. And then C doesn't really have anything to do with how fast it moves through. This would move through just as fast as these pieces. I know what you were thinking for C. You were thinking that meant you absorb it faster or something, weren't you? Which isn't what it says. So it's one of those answers that you're reading it and you're like, I think that's what it means. But it doesn't. So yeah, you could also say, it helps you absorb lipids faster. You could say that. Right. All right, and I think, let's see, I don't think this is on the study guide if I remember right. But I just wanted to show you up there, your small intestine also makes a whole bunch of its own enzymes. So it has the enzymes from the pancreas, and then it makes all of these as well to break down, well, I guess one thing that's different, like maltose, sucrose, and lactose, those are all dipeptides. So we made the amylase to break down the bigger pieces, and now we're breaking it into the final little pieces. And there's your lactase there. How many intolerant people are here? Lactose intolerant, not some other intolerant. Anybody? Nobody? There's actually a strong correlation with what your ancestry is. So if your ancestors were more shepherds and lots of milk in the diet, you're way less likely to be lactose intolerant. So interesting. So I don't know if you remember learning about simple diffusion, facilitated diffusion, and active transport in 201 a little bit. You probably hated it. It's okay. So just wanted to point out that all of those processes are happening in the small intestine. So you've got certain things that are absorbed by simple diffusion. Over here is the lumen, like inside the intestine. Over here is like the blood vessels, the capillaries. So the blood's flowing past. The digestive stuff is flowing past here. So you've got amino acids going by diffusion. The fatty acids that you broke down, they're coming in through the diffusion. And you also have some things going in through active transport and facilitated diffusion as well. So you're using all three processes. Just wanted to kind of point that out. All right, and one interesting fact that people may not know is so here's the intestine, like the villi sticking up in the small intestine. It's the same for the large intestine too. So blood's coming in and it's picking up all of the nutrients. And then from there, it goes through the hepatic portal vein to the liver. So your liver is the first organ that sees all the nutrients you absorbed. And that's important because it can kind of modify them or store any extra ones and perhaps filter out any really bad toxins that may have ended up there. And then from there, it goes through basically back to the heart to get pumped to the lungs, and then to the heart, and then out to the rest of the body. So like a nutrient absorbed to get to like your bicep, you would have to go to the liver, to the heart, to the lungs, to the heart, and then to your muscles. So it's quite a long thing there. So it's right there. So it goes into the sinusoids, the central vein, the hepatic vein, and right here it should say liver, which is weird it doesn't. And then inferior vena cava and the right atrium, for being specific about where all those things are. All right, before we do that question, if you look on like labels of certain drugs, what is the side effects they usually mention, or what organ is most often mentioned? Yeah. So why do you think that would be? Yeah. So any sort of oral thing you're taking, the liver is going to be exposed to it first, and also at the highest concentration. So it hasn't been like, diffused out into the whole bloodstream. It's just right from the intestine to the liver. Which a lot of the times, if you have to get there's certain antibiotics and drugs that you have to get IV. A lot of the times that's because if you ingested it, it'd be too toxic to the liver. So instead they can just inject it in the blood, and it doesn't end up at such a high concentration in there. That's not the only reason, but that is one of them. All right. So let's do some stuff with your group. Question 1. All right, so let's go ahead and keep going. I'm sure you're only supposed to do question 1 for now. It's okay, we'll get to 2 soon. All right, so what about all the hormones involved with the enzymes? But now we need the hormones to help us control everything. So we're going to go through these sort of quickly and then have you do some practice with them. So the first hormone is gastrin. What was I going to say? Oh, so if you remember, what are the 4 main parts of a feedback system? All right, so we've got a stimulus. That's first. And what does the stimulus activate? The receptor. And what does the receptor? The receptor tells something. Control center. And then the control center tells the effector or effectors, possibly. So for all these different hormones and loops, they're going to have those same 4 components. All right, so gastrin, as you saw in your study guide, it's made by the G cells. And the stimulus is either a high pH of what's in the stomach or the stomach stretching. And the response is it tells the parietal cells to make more HCl, which, I mean, kind of makes sense. If the pH is too high, what do you need to do?
Base it.
What?
Make it more basic.
No, it's too basic, so you want to make it more. Sorry, I know where you're getting stuck.
Never mind.
I know.
What you're saying. You want to kill
So
those dirty germs in your
acids down by 0 or 1, base is here, but then when it gets confusing is high pH is actually up here, and low pH is down here. So I think people always want to say high pH is acidic, right? Just because it sounds, I don't know, you're thinking about the H pluses probably. So anyways, high pH is basic, so you need to make more HCl. Or if your stomach is stretching out, what does that mean? Why do you need more HCl then? Yeah, well, if you just ate something, your stomach's going to stretch, so you probably need more HCl, exactly. So it can be activated by either one or both. All right, and just so the other cell types in there, we did the G cell. And if you notice, it's kind of at the bottom of the pit that lines the stomach. So your stomach isn't completely flat. There are these sort of microscopic pits in there. So you got the G cells. The chief cell makes lipase as well. Parietal cell makes HCl. Mucus cell makes mucus. And you've got other mucus cells as well. There's the different cell types. So here's your feedback diagram for the stomach, control of stomach acid that we just talked about. So the stimulus, high pH or stretching the stomach. The receptor would be either the chemoreceptors that are in the G cells or stretch receptors in there. In the control center is a little cluster of neurons called the submucosal plexus. So it's not up to the brain or anything. There's actually some neurons that receive information and decide what to do about it, all the way down by the stomach. Part of the parasympathetic nervous system. And the 2 effectors are the parietal cells making HCl, and the one we didn't mention yet is it tells the smooth muscle to contract more and like stronger and more often to help the mechanical digestion of the food. And so that's going to result in more acid, more mixing, faster digestion, which will fix both of those stimuluses. Because if you're digesting it faster, then the food's going to get to the small intestine faster, and the stomach won't be stretched anymore. So it's kind of a good system to not make extra HCl. Once the stomach's empty, you don't need to make extra HCl because it's done. All right. So next up is question 2, which is true and false, and question 3, which is about that diagram, feedback diagram on the back, which I'll put back up here. All right, so as you're working on number 3, Make sure at some point in your answers, you're talking about what symptoms the person would have. In addition to whatever else you write down, but that should be part of it. Like, all right, let's keep going. So anyways, that's good. Those are good practice questions for what we're going to be doing in here. So you got to look at those diagrams, and you kind of have to connect. Like, well, if this breaks, then this is going to happen, and the person's going to feel this. You kind of have to put both those pieces together. All right. So the next two, so we got, I'll get 2 minutes. So our next two hormones are secretin and cholecystokinin. Once again, the same kind of steps. For secretin, the stimulus is low pH of chyme coming to the small intestine, and the response is secretion of bicarbonate by the pancreas. And then the second one is the cholecystokinin. Stimulus is that you have not fully digested macromolecules in the small intestine, and the response is the secretion of pancreatic juice and bile. So what you're going to do is draw the feedback loops for those two hormones on your table with your group. And pretty much almost all the information you need is on that slide. So think about what's the stimulus, what's the receptor, and what's the effector. See what you could do. Oh, so if you're stuck, think about, you can look at that stomach acid diagram, kind of get an idea of, the arrows and stimulus, whatever. Cool. Well, y'all can head out then. We'll get the rest of these folks done. All right. Sweet. Sweet. Sweet.